So let's start our discussion of a very important and a very basic topic, which is cell physiology, and this will be the only video that will cover the whole chapter. So this chapter is often well neglected, so इसको आप neglect नहीं करना and give this topic due time and importance, because 2 to 3 questions are expected from this topic in your exam, and this is equally important for every postgraduate entrance exam, जिसमें ये है कि you need to go through this ताकि ये कि आपकी basic चीज़ें आपकी revise हो जाएं. So I'll try to cover all these topics. So timestamps will also be provided, so you can simply go to your topic जिसमें आपको confusion है, और otherwise I'll recommend कि आप एक दफ़ा go through कर लें इन सब topics को. So let's just start with the first topic, which is cell membrane. तो cell membrane के बारे में you must know that it is primarily composed of phospholipids and proteins. So this right here is a very basic structure of the cell membrane, lipid bilayer. So lipid bilayer is composed of a glycerol backbone which contains these hydrophilic heads. Each head is attached to 2 hydrophobic fatty acid tails. So this is the hydrophilic head and 2 fatty acid tails. These tails face towards each other. That is why it is known as a bilayer. So this is basically a bilayered structure. So as it is a lipid bilayer, so just recall your concept of like dissolve like. So anything which is lipid soluble, it can pass through this membrane easily. Examples क्या हैं? Like oxygen, like carbon dioxide. और यहाँ पर एक important high yield point add कर दूँ, that later on we will study the mechanism of action of steroid hormones. तो वो they act on the nucleus. So nucleus में जाने के लिए they have to cross this membrane. तो because they are lipid soluble, so they can easily cross this membrane and enter the cell. So lipid soluble substances can pass through this membrane easily. And what about water soluble? So water soluble substances, they cannot pass because they are lipid insoluble. So example में you have charged ions like sodium ion, chloride ion, even water and glucose. So they need specific or specialized transporters to move across the membrane. So they cannot cross this cell membrane. इसके अलावा in the structure of cell membrane there are certain proteins. अब कुछ proteins हैं which are embedded or anchored to this membrane. So these proteins are known as integral proteins. Integral proteins, they are embedded or anchored to this membrane, and they are anchored via hydrophobic interactions. So these are the transmembrane proteins, like they cover the almost entire length of this membrane, and they usually work as the ion channels and specific transporters. There are other proteins as well that are known as peripheral proteins. These peripheral proteins, they are not embedded, not anchored, rather they are loosely attached, and they do not cover the entire length, may be present just like here. So these proteins, they are attached via electrostatic interactions. So let's say we have 2 cells, cell number 1 and this is cell number 2. So there must be a way कि जिसमें ये दोनों सेल्स आपस में कम्युनिकेट करें, आपस में इंटरेक्शंस करें. सो यहाँ पे 2 सेल्स के दरमियान में इट इज़ नोन एज द जंक्शन. दीज जंक्शंस कैन बी ऑफ डिफरेंट टाइप्स. लेट्स से वन ऑफ इट इज़ टाइट जंक्शंस. अब टाइट जंक्शंस हर उस जगह पे होते हैं जहाँ पे लिमिटेड इंटरेक्शन रिक्वायर होते हैं 2 सेल्स का आपस में, और एग्जांपल इसकी मिलती है एपिथीलिया में. These tight junctions are also known as zona occludens. So this question has been asked previously in the exam. So tight junctions are also known as the zona occludens. So there are two types of tight junctions. One is an impermeable one, कि कुछ भी हो जाए ये communication allow ही नहीं करते, and some are partially permeable or leaky. So example of the impermeable tight junctions or impermeable zona occludens is the DCT of the nephron. And examples of the leaky tight junction is PCT of the nephron or gallbladder. One other example of such type of junctions is a gap junction. Gap junction is present between the cells where a lot of communication is required, so they permit communication. Example is myocardial cells. Moving on to the next topic, which is cell organelles. So first up we have smooth endoplasmic reticulum. Smooth endoplasmic reticulum, the name is smooth. It means that there are no ribosomes attached to the surface. दूसरा high yield point, it is the site of steroid synthesis. तो steroid synthesis अगर सेल में होगी तो it is in the smooth endoplasmic reticulum. Another important function is detoxification of drugs or harmful chemicals or toxins. तो steroid synthesis कहाँ पे होती है जनरली हमारी बॉडी में? एड्रीनल कॉर्टेक्स में, गोनेड्स में. सो द एड्रीनल कॉर्टेक्स एंड गोनेड्स, इसमें स्मूथ एंडोप्लाज्मिक रेटिकुलम की जो स्ट्रेंथ है या फिर नंबर ऑफ द ऑर्गेनेलीज हैं ये बहुत ज़्यादा होती हैं. सिमिलरली डिटॉक्सिफिकेशन ऑफ ड्रग्स इज़ अ फंक्शन ऑफ लीवर. सो लीवर इज़ ऑल्सो एन ऑर्गन विच इज़ अबंडेंट इन स्मूथ एंडोप्लाज्मिक रेटिकुलम. दिस स्मूथ एंडोप्लाज्मिक रेटिकुलम इज़ ऑल्सो द लोकेशन ऑफ glucose 6 phosphatase. So the details of this we will study in biochemistry lecture, but ये याद रखें कि glucose 6 phosphatase is very important in the last step of gluconeogenesis. Similarly, it is also important in the last step of glycogenolysis. So moving further to the rough endoplasmic reticulum. So by rough we know that ribosomes are attached. Apart from that, it is the site of secretory proteins. तो जहाँ पे because it has ribosome attached, इसका मतलब है प्रोटीन सिंथेसिस यहाँ पर हो रही है. बट यहाँ पे कौन सी प्रोटीन्स बन रही हैं जो कि सेल ने सिक्रीट करनी है आउटसाइड टू परफॉर्म द फंक्शन. सो ऑल द सिक्रीटरी प्रोटीन्स दे आर सिंथेसाइज्ड इन द रफ एंडोप्लाज्मिक रेटिकुलम. फॉर एग्जांपल plasma cells that are going to secrete antibodies. Rough endoplasmic reticulum is also responsible for the synthesis of the proteins which will remain within the organelles, like endoplasmic reticulum itself, Golgi apparatus, and lysosomes. But ribosomes, which is in fact the factory of protein synthesis, it is responsible for the release of cytosolic proteins or mitochondrial or peroxisomal proteins. So just memorize कि peroxisomes की, माइटोकॉन्ड्रिया की, साइटोसोल की प्रोटीन्स दे आर सिंथेसाइज्ड बाय द राइबोसोम्स endoplasmic reticulum, Golgi apparatus and lysosomes की proteins are synthesized by the rough endoplasmic reticulum. Protein synthesis is the function of ribosomes, but if ribosomes is not in the option, you can click rough endoplasmic reticulum because it has the ribosomes attached. Golgi apparatus, just इसको ऐसे देखें जैसे you are looking at the working of a factory. वहाँ पे प्रोसेसिंग ऑफ प्रोडक्ट है, पैकेजिंग ऑफ प्रोडक्ट है, लेबलिंग ऑफ प्रोडक्ट है एंड डिलीवरी ऑफ प्रोडक्ट है. तो ये सारे काम गोल्जी अपरेटस के हैं. सो द प्रोक्सिसोम्स they originate from स्मूथ एंडोप्लाज्मिक रेटिकुलम. सो इसमें कुछ ना कुछ शेयर्ड फंक्शंस भी होंगे. सो दिस प्रोक्सिसोम्स दे कंटेन catalases, oxidases and hydrogen peroxide. So talking about the functions of the peroxisome. First function, beta oxidation of very long chain fatty acids. It is strictly the function of peroxisome. Next, alpha oxidation of branched chain fatty acids. It is also a strict function of peroxisome. Third, that you need to memorize is it is involved in the catabolism of amino acids and alcohol, ethanol. The peroxisomes, they are also involved in the synthesis of bile acids and also the plasmalogens. These are the important membrane phospholipids present in the white matter of brain. That is why जब इसकी डेफिशिएंसी होती है या अगर ये काम ठीक से ना करे तो हमें न्यूरोलॉजिकल साइन एंड सिम्टम्स भी आते हैं. So one important syndrome associated with it and asked multiple times in the exams is Zellweger syndrome. So it is basically a reduction or complete absence of peroxisomes. तो इसमें साइन एंड सिम्टम्स में वी हैव हाइपोटोनिया, सीजर्स, jaundice, craniofacial abnormalities, hepatomegaly leading to death. So the next step is proteosomes. अब proteosomes का क्या काम होता है? It is basically a barrel shaped protein complex जो कि डिग्रेड करता है मिसफोल्डेड प्रोटीन्स को. सो बेसिकली ये जो सारी लेबलिंग प्रोसेसिंग ये हमने याद किया था कि इट्स अ फंक्शन ऑफ गोल्जी अपरेटस. सो अगर गोल्जी अपरेटस से एक प्रोटीन रिलीज हुई है विच इज़ मिसफोल्डेड तो उसको सेल क्या करता है, टैग कर देता है विद ubiquitin. So this is a signal for proteosome to detect that protein and degrade. So if I ask you misfolded proteins from the Golgi apparatus is degraded by, so the answer should be proteosomes. So next up we have cytoskeletal elements. These are basically network of proteins within the cell or within the cytoplasm to support the cell. So it support the cell structure and also involved in the movement of cell and cell organelles, also involved in cell division. So different types होती हैं इसकी. One is microfilaments. These microfilaments like actin, microvilli, they are involved in the muscle contraction. एक तरह की एग्जाम्पल है इंटरमीडिएट फिलामेंट्स. सो इंटरमीडिएट फाइबर्स जो हैं ये अब हैं कौन से, ये मेमोराइज करना है. विमेंटिन, जीफैप, न्यूरोफिलामेंट्स and cytokeratin. So they are all intermediate filaments. So GFAP is basically glial fibrillary acidic protein. And one last example is microtubules. So microtubules में we have cilia, flagella, mitotic spindles and centrioles. So one common exam question, what are the drugs that acts on the microtubules like cilia, flagella, mitotic spindles and centrioles? So to memorize this, we can use a mnemonic which is microtubules get constructed very terribly. So with this M, we have mebendazole, which is an antihelminthic drug. With this G, we have griseofulvin, which is an antifungal drug. This C is for colchicine, which is an anti-gout. Vinca alkaloids and T for taxanes, both of which are anti-cancer drugs. So these microtubules have a positive end and a negative end. So this positive end, P for positive, P for periphery, so it is towards the periphery, and the negative end is towards the nucleus. So one important thing about these cilia, so there are motile cilia which consist of 9 microtubules, doublets, along with 2 microtubule singlets, and the basal body, which is in fact the part of the cilia which is below the cell membrane, it consists of 9 microtubules triplets. So there is a disease known as primary ciliary dyskinesia, in which there is an immotile cilia. So cilia is very important क्योंकि जहाँ पर भी बॉडी में मूवमेंट रिक्वायर्ड है सिक्रीशंस को आउट करने के लिए, वहाँ पे सीलिया प्रेजेंट होते हैं, लाइक द रेस्पिरेटरी ट्रैक्ट, लाइक द जेनिटो यूरिनरी ट्रैक्ट. So one of the syndrome that is associated with primary ciliary dyskinesia is Cartesian syndrome. So Cartesian syndrome has primary ciliary dyskinesia and also sarcomere is basically a basic unit of contraction of the muscles. So which muscles? Skeletal and cardiac. No smooth muscles. Smooth muscles don't have sarcomeres, so no striations, hence known as the smooth muscles. So this right here is a sketch of a relaxed muscle fiber. So in a relaxed muscle fiber, the part between the two Z lines is known as a sarcomere. This sarcomere has these thin filaments and these thick filaments. So when we talk about the thick filaments, they are present in the A band. So this is the A band. So thick filaments are present along the entire length of the A band and primarily composed of myosin. The thin filaments, they are anchored to the Z line in the region of I band. So here the thin filaments are anchored to the Z line in the region of I band and they contain actin, tropomyosin and troponin. These troponin are of three types: troponin I, troponin C and troponin T. This C is for calcium. It binds to the calcium and permits the interaction of actin and myosin. This troponin T, it attaches this troponin complex to tropomyosin, and this troponin I is for inhibition. So these thin filaments, they interdigitate with the thick filaments in the region of the A band. Along the actin myofilaments, there are certain binding sites for the myosin heads, and the myosin head can attach to these binding sites and can generate the force for the contractions. So simply, the force of contraction is the function of myosin myofilament or the thick filament. The only filament which is present in the H zone is thick filament. So now we'll move to the fully contracted muscle. So if we compare the both, here you can appreciate the H zone, but here the H zone has disappeared. So while looking at the contracted muscle, the H zone shortens or disappears. The I band, first it was of this length and now it has decreased its length. So the I band, it also shortens. But the A band, it has remained unchanged. Not even for the charge but also for the size. So sometimes these channels may be open and allow the movement, and other times they are closed, so no ions or no movement of the selective charge or selective particle. Now we will continue our discussion on the action potential. So first we need to understand the functioning of the ion channels. The ion channels are basically the integral proteins that we have understood already, that they are present along the entire length or entire thickness of the cell membrane, and they allow the passage of the substances which cannot easily pass through the cell membrane, that is the water soluble substances, or simply you can say the sodium ions. So they permit basically ion channels, ion movements. So they are the ion channels. These ion channels are highly selective. So these terms of voltage gated and ligand gated you will hear multiple times. So you need to understand that the voltage gated channels, they open or close by the changes in the membrane potential. While the ligand gated channels, they open or close by the interaction of certain hormones, neurotransmitters, or second messenger. So these ion channels have activation and inactivation gates. If both of the gates are open, then we can say that the channel is active and it is open. If either of it is closed, we can say that the channel is closed. So if the inactivation gate is closed, then we can say that the channel is closed and inactivated. And if the activation gate is closed, we can say that the channel is closed and is resting. Now we'll see the effect of depolarization on these gates. So the activation gates becomes open when there is a depolarization, and at the same time this depolarization leads to the closure of the inactivation gate. So if the inactivation gate is closed, we can say that the channel is inactivated. Hence, during the depolarization phase, no new action potential can be generated. So resting membrane potential is basically a measured potential difference across the cell membrane, and by convention it is measured in millivolts. It is basically the intracellular potential relative to outside. And so if we can say that if there is a resting membrane potential of minus 70 millivolt, it is 70 millivolt cell negative. So what contributes to the resting membrane potential? Is it the sodium potassium pump, or is it the diffusion potential? So the answer to this question is diffusion membrane potential, because the sodium potassium pump contributes only indirectly. The contribution of the sodium potassium pump is small. So diffusion membrane potential is basically result from concentration difference of the permeant ion. So whenever there is a stimulus, it may not always lead to the action potential unless a threshold is reached. So after a threshold is reached, action potential is self-sustainable or inevitable. So a few terms. One is depolarization. Depolarization simply means making the membrane potential less negative. So less negative का मतलब है कि as we have already understood that it is actually the intracellular potential difference relative to outside, so it has to make intracellular less negative. And hyperpolarization is basically making the membrane potential more negative. It means that making the intracellular compartment more negative. So whenever we are talking about the currents or the movement, we always take positive charges. So if the positive charges are moving outside, if the positive charges are moving inside, so we can say that it's an inward current. And if the positive charges are moving outside, so we can say it's an outward current. And one last thing about action potential, that it follows all or none principle. So this right here is a simple diagram of the action potential. This A is the resting membrane potential, and if at all a stimulus is given which does not reach this E, which is the threshold, so it will result in a failed attempt. So once this threshold is reached, the action potentials become inevitable and there is an upstroke of the action potential, and this B is known as depolarization, and by depolarization we mean that the positive charges are moving inside and making the cell less negative from the inside. And this C right here is repolarization, and it is due to the movement of the positive charges outside or the outward current. This D right here, this one, this is hyperpolarization. So now we'll talk something about refractory period that is important for the exam or from the exam point of view. Simply, there can be an absolute refractory period or a relative refractory period. Absolute refractory period means when no action potential can be elicited no matter how large the stimulus is. And in the relative refractory period, action potential can only be generated if the stimulus is stronger than the usual. In a nutshell, we can say that whenever there is an action potential which is already existing, the entire period of action potential is absolute refractory period. So now we will discuss transport across the membrane. So we'll just simply go through this table which is given in the PRS physiology. So first up we have simple diffusion, and simple diffusion is always downhill or down the concentration gradient. It means that if there is a simple diffusion, then the substance is moving from a higher concentration to lower concentration and requires no additional thing, like no carrier is required, no energy is required, no sodium gradient is required. Similarly, in the facilitated diffusion, the only difference here is the presence of a carrier. So whenever there is a presence of carrier, it means that there are certain limitations that are involved. So next is primary active transport. Primary active transport is basically the usage of energy. So whenever we are using energy, we are using a carrier, then we are moving the substance from a lower concentration to a higher concentration. So the next is co and counter transport, and both are examples of secondary active transport. So co-transport means that the substance is moving along with another substance in the same direction, and counter transport means that a substance is moving along with another substance in the opposite direction. So it is always uphill, and it requires energy, but this energy comes indirectly. अब वो कैसे, इसको अंडरस्टैंड कर लेते हैं. लेट्स सपोज देयर इज़ अ को ट्रांसपोर्ट में ये होता है कि बोथ सब्सटेंसेस आर मूविंग इन द सेम डायरेक्शन. सो लेट्स से द अदर आयन इज सोडियम. सो सोडियम इज़ मूविंग इनसाइड, और उसके ग्रेडियंट की वजह से जो एनर्जी प्रोड्यूस हुई है ये यूज़ करता है हमारा आयन टू मूव इन द सेम डायरेक्शन. सो इन काउंटर ट्रांसपोर्ट लेट्स से दैट सोडियम इज़ मूविंग इनसाइड ऑफ द सेल and our substance is moving outside of the cell, let's say potassium. So now the cell will move outside of the cell with the energy that was produced by the movement of sodium along the gradient. Now we simply highlight some important points that are related to the transport physiology. 1. Simple diffusion is the only form of transport that does not require any carrier. Rest all require carriers. So basically this is diffusion, and diffusion is equal to PA C1 minus C2. This C1 minus C2 is the concentration gradient. So it means that diffusion is directly proportional to the concentration gradient. This A right here is for area, so diffusion is directly proportional to the area as well, and P stands for permeability. And this permeability is basically the ease with which a solute diffuse through the membrane. This permeability is dependent on various factors, and one important one is oil-water partition coefficient. So this permeability is directly proportional to it, and if the oil-water partition coefficient is high, then there is more diffusion, more permeability, eventually leading to increase in the diffusion capacity. Next is size of the solute. Size of the solute is inversely related to permeability. So if the size increases, the permeability decreases, leading to decrease in diffusion. The next one is membrane thickness. So membrane thickness is also inversely related to permeability. So if there is an increase in the thickness, there will be a decrease in the permeability, and there will be a decrease in the diffusion capacity. If we compare simple diffusion and facilitated diffusion is more rapid than simple diffusion. And recall from the knowledge of biochemistry, there are two isomers of glucose, D-glucose and L-glucose, levo and dextro. This D-glucose can be transported via facilitated diffusion, and this L cannot. Moving on to a very important topic, which is neuromuscular junction. Same, this diagram is also taken from the BRS physiology. So you just understand the sequence of events that are taking place along the neuromuscular junction. So first up, we have action potential in the nerve. So first of all, there will be a synthesis and eventually storage of acetylcholine in the presynaptic terminal. So this is presynaptic terminal, this is synaptic cleft, and this is postsynaptic terminal. So first step, there will be a synthesis and storage of the acetylcholine in the presynaptic terminal. There comes the action potential, and when the action potential reaches here, there will be step 2, which is opening of the calcium channels and movement of the calcium inside. So in the second step, there will be depolarization of the presynaptic terminal, leading to opening of the calcium channels. In the step 3, these calcium channels will cause the release of acetylcholine in the synaptic cleft. So in step 3, after the calcium uptake, there will be release of acetylcholine in the synaptic cleft. Fourth step is the diffusion of acetylcholine towards the acetylcholine receptors. So in fourth step, there is diffusion of acetylcholine towards the acetylcholine receptors, and eventually it will lead to the depolarization of the motor end plate, and there will be action potential in the muscle, leading to depolarization of post synaptic terminal. So just memorize this sequence. This sequence is asked in the exam. So next up we have excitation contraction coupling. So excitation contraction coupling को समझने के लिए first of all you need to know what is a T-tubule. T-tubule is a part in the muscle fiber which is in direct communication with the extracellular compartment. तो यहाँ पर हो क्या रहा है कि सबसे पहले in step 1 there is action potential that enters from the adjacent cell. तो दूसरे सेल से action potential move करता हुआ इस सेल की तरफ आ रहा है and it will move across this T-tubule. So यहाँ पर हैं L type calcium channels and these calcium channels, once the action potential reaches these channels will become open. So voltage gated calcium channels open and calcium moves inside of the cells. So once inside of the cells, calcium channel moves towards the ryanodine receptors which are present in the sarcoplasmic reticulum. Sarcoplasmic reticulum is basically an organelle in the muscle fibers which is the reservoir or storage of the calcium. So in step 3, calcium release through the ryanodine receptor channels. So the calcium will be released and it will move in the cytosol, and here it is known as the calcium spark, as written here that in step 4 calcium release causes calcium spark. So in step 5 there is a summation of the calcium spark. All the calcium, they sum up and create a calcium signal. So this calcium signal will move towards the sarcomere and it will bind to the troponin C, as we have already discussed in the sarcomere topic. So in step 6 there is calcium binding to the troponin to initiate the contraction. So the reverse steps for the relaxation. Here relaxation occurs when calcium unbinds from the troponin and calcium is pumped back into the sarcoplasmic reticulum. Now this excess calcium is exchanged with the sodium by the sodium calcium antiporter or counter transporter. So this is all you need to know about the excitation contraction coupling that is taking place in the skeletal muscle fibers. Moving on to the muscles. So we'll just use this table to identify what are the key differences and key similarities in the skeletal, smooth and cardiac muscles. So first of all we will discuss about the appearance. As we have already understood that smooth muscles, they are known as smooth muscles because they have no striations. Why they have no striations? Because they have no sarcomeres. So the sarcomere is only present in the cardiac muscles and the skeletal muscles, hence they are striated. So second up is the upstroke of the action potential. Upstroke of the action potential in the skeletal muscle fibers is the invert sodium channels, while in the smooth muscle we'll also discuss this in GIT that there will be an invert calcium channel. So the details of the action potential of the cardiac muscles will be discussed in the CVS topic, but here you just need to know that the SA node follows the invert calcium current for depolarization, and all the other components of the cardiac muscles like the atria, the Purkinje fibers and the ventricular muscles, they are depolarized by the invert sodium channel, sodium current. Plateau phase is only present in the ventricles, atria and the Purkinje fibers due to the invert calcium current. So just in the step 2 there will be a plateau phase, and this is due to the invert calcium channel. Duration of action potential, it is the quickest in the skeletal muscles and moving, and you can appreciate others as well. So excitation contraction coupling, we have discussed in details about the skeletal muscle and the smooth muscle. The action potential opens the voltage gated calcium channels in the cell membrane. So it is kind of the same, but with the addition of the hormones and transmitters through the IP3 pathway. So this one is the most important thing, that molecular basis for the contraction. The molecular basis for the contraction in the skeletal muscle is calcium troponin C complex, as we have already discussed in the previous topic of the sarcomere, that troponin is a complex of 3 proteins: troponin I, troponin C and troponin T. And in the smooth muscle there is a calcium calmodulin complex which increases the myosin light chain kinase activity. So just memorize this. And the contraction, molecular basis for contraction in the cardiac muscle is same as that of the skeletal muscle. Now we'll talk about permanent, stable and labile cells. So what are permanent cells? First of all इसकी examples देख लें. Neurons, they are permanent cells. Skeletal muscles, they are permanent cells. Red blood cells, permanent cells. Why so? Along with the skeletal muscles, smooth muscles also are permanent cells. So why they are known as the permanent cells? Because they always and always remain in the G0 phase of the cell cycle and cannot move to the G1 phase. What are stable cells? They are present in the G0 phase, but they can move to the G1 phase when required or stimulated adequately. Example is liver cell or hepatocytes. Other examples include lymphocytes, PCT cells, and periosteal cells. Labile cells always and always remain in the G1 phase, and this G1 phase is very short as well for the labile cells, and they never ever go to G0 phase. These are the cells which are most affected by chemotherapy because they are highly rapidly dividing cells. And examples include bone marrow, certain all the epithelias, skin, hair, and germ cells. In the next lecture, we'll discuss all the important one-liners from the cell physiology. So consider subscribing to the channel so that the new videos can reach you timely. Thank you so much.